I'm Clémentine Schiltz, and I'm very pleased to be here today for this live session from the MOOC Viral Outbreaks and Pandemics. I'm the head of digital education at Institut Pasteur in Paris. And I would like to thank our directors, Pierre-Emmanuel Secaldi, Jean-Pierre Vartagnan, Arnaud Fontanet, and Stéphane Ziantara, and our community manager, Laurie Pinault, who coordinated this MOOC. I would like to invite all of you to present yourself in the discussion. And for example, tell us your first name, last name, and where you're connected from. And also, of course, we will start to address the questions that were asked in advance, but then we'll ask your question, your live questions. And for this, I'll ask you to use the Q&A button on the right of your right-hand corner of your computer. So please present yourself, and Laurie, the floor is yours now.
Thanks, Clémentine. So I'm Laurie Pinault. I'm the community manager for this MOOC. So we interacted a bit together on the forum of the Fun platform, and I was sending you the emails each week. So I will ask the different co-directors to just briefly present themselves so you identify who's who, and then we can start with the questions that some of you asked in the registration form for the live session. So maybe Arnaud or Stéphane, I don't know who wants to start.
Okay, so good afternoon. My name is Arnaud Fontanet. I'm the head of the Emerging Disease Epidemiology Unit at Institut Pasteur. I'm also professor of public health at the Conservatoire National des Arts et Métiers.
Hello, I'm Stéphane Ziantara. I'm the vet of the group. I'm also a virologist, and I'm director of the animal health in ANSES in Maison Alfort, in the campus of the vet school in Maison Alfort near Paris.
Hello, I'm Pierre-Emmanuel Secaldi, professor at Université Paris-Cité, and I work in the oncogenic viruses unit in Pasteur Institute.
Good afternoon, my name is Jean-Pierre Vartagnan. I'm research director at the Institut Pasteur. I'm working in the Department of Virology, and I'm at the head of the unit Viruses and Cellular Stress.
Thanks to all of you. We'll start with the questions that were asked in the registration form for the live session. So there were multiple questions asked by learners on past and future pandemics. So could you summarize why do we witness an increase in the occurrence of viral pandemics in the past decades? What is the risk for a new pandemic to occur in the future? And can we predict somehow what it could be? So, Stéphane, do you want to start, maybe?
Okay, yes, so I will try to answer to these questions. In fact, there are different that have occurred these last decades. Probably one of the first is international trade. You have an increase in international trade of people and animals. As a vet, I can tell you that you have a lot of domestic animals which are able to cross the world, and of course, the human beings and animals can be infected with different pathogens, and the SARS-CoV-2 is a very good example of the virus emerging somewhere in the world and being disseminated all around the world. So international trade of products. I've got an example of a veterinary disease, which name is blue tongue, transmitted by culicoides in the north of Europe. And probably the fact that you have an increase in the flower trades. You can offer trades to your mother-in-law coming from Kenya or other countries around the world. And these flowers will come to Europe, for example. You will have larvae, you will have insects, and these larvae or insects can be infected with different viruses. You have also all the destabilization of society, the war, for example, which have consequences on the absence of human and veterinary surveillance diseases. You have also the global warming, which has also an important role. First of all, by the destabilization of the society with people leaving the countries. and also with vector-borne disease, for example. And we all know that in many countries around the world, you have arthropods which were not present, which are now present in these countries. If you take the example of Europe, in 2022, there were 66 cases of dengue, autochthonous dengue virus in the south of France. In 2023, there were cases of dengue not far from Paris. So we really see a modification of the epidemiological patterns of many diseases because these vectors find now in our countries or country which had more colder weather, the conditions to develop, which is a new pattern that we see now. So this is some examples of the factors of emergence. And to predict which will be the next emerging disease, it's really difficult. I will not try to do this exercise because I know many diseases nobody has ever imagined that they could emerge in the vet world, but also in the human world also. It's really difficult. Of course, there are diseases, and probably we'll talk about the situation in a few minutes, which are under surveillance because they are really at risk of expansion. But to really predict what will be the next, personally, I will not try to give a precise name of such disease. So I don't know if Arnaud, you want to complete what I said.
I would just add to give some perspective. We really have the feeling that there is an acceleration in this phenomenon of emergence of infectious disease. It's not only that we are better capable to detect them, but if you just consider the last 40 years of the 20th century, we had the emergence of HIV/AIDS, which was a major pandemic. We also had Ebola, for instance, identified for the first time in 1976. But that's basically it. If you look for the main ones, I mean, if you look at the first 20 years of the 21st century, we saw SARS, we saw pandemic flu in 2009, we saw MERS in 2012, we saw Zika in 2015, we saw COVID-19 in 2019, Mpox in 2022. And I'm just telling about the big ones, you know, I mean, there are plenty more, but we have already had 5 very significant emergence in the beginning of the 21st century. And that comes in line with what Stefan was just explaining. You know, there are many different factors that facilitate the emergence of infectious disease. And Stefan is perfectly right in saying that he would not want to try to predict because we have always been taken by surprise. I mean, the one which I say was a bit a similar scenario is COVID-19. After all, we have the story of live markets in China and virus coming out the same way that we had SARS in 2003. But who would have predicted that MERS, for instance, a beta coronavirus, would emerge from dromedary camels in the Gulf countries in 2012? Totally unexpected. Who would have thought that the Zika virus, which was living quietly in Africa and Southeast Asia, would decide to cross the Pacific Ocean and starting in 2005, 2007, and reach Latin America in 2015. You may remember these terrible pictures of children born with microcephaly from the infection of pregnant women by this Zika virus. Ebola, which used to be in Central Africa, ended up with a massive epidemic in West Africa in 2013, 2014. And even the pandemic flu in 2009. I mean, we used to say that this pandemic flu would eventually start from Asia, particularly China, but here it was detected first in Veracruz in Mexico. So telling which is going to be the next one, where it will emerge, and when is something that we are just not capable of doing today. And this means also we have to be very prepared for the surveillance so that we are capable of detecting outbreaks at the very beginning, because it's probably at the very beginning that we have the best chance to contain a starting outbreak. And this is the goal of all those surveillance systems that have been put in place in part coordinated with the Global Outbreak Alert Response Network of WHO for human disease. And maybe you could complete a little bit, Stefan, for the surveillance of veterinary disease. I mean, how is it organized? And so that we can detect very quickly whatever would emerge from the animal world?
Yeah, just one point perhaps to complete what you said, Arnaud, is that you have a lot of viruses, as you have said, and the majority, not all of us, but, not all of them, but the majority were RNA viruses. This is probably one point because they have a high variability, genetic variability, and they are able to adapt and to cross the species barrier, for example. And I'm not saying this is only RNA viruses. Monkeypox is a good example that it is not RNA viruses, but in general, we have this point. And concerning the veterinary surveillance system, it's more or less organized as in human system. So of course, we try to have as many collaboration as possible with countries which are in the neighboring of Europe, for example. And then in different countries, we have private vets who are working for the state, so they can have an official mandate to sample the animals when they find some clinical signs which are a bit different from what they are used to see. We have a network of laboratories. We have veterinary services, which are state organization to prevent the diseases, to fight against the diseases. So more or less the system is quite similar to what you can find in the human organization.
Thank you. I think you started to tackle one of the questions that was prepared for a bit later. So going on then with surveillance, are there particular identified pathogens at risk of emergence that are currently under surveillance with the different systems that you just described, Arnaud and Stefan? Or yeah, should we have some particular pathogens in mind that are under surveillance?
I may start. There are definitely some pathogens we are very concerned about. I mean, all what goes around influenza and the associated disease is flu. It's very well known for its capability to give large pandemics because first, it's a respiratory virus. We all breathe, so we are all at risk. It's not dependent of whether you are close to a certain vector or animal. I mean, we know that it's a pathogen that can circulate widely very quickly. And influenza are also very difficult to stop because they tend to be contagious before onset of disease. And therefore, all our control measures around isolation and quarantine do not work very well with influenza. And this explains why whenever we had new influenza viruses that resulted from reassortment of genomes across different types of influenza viruses, the new ones can be responsible for very large pandemics. And we had 3 major examples in the 20th century with these so-called Spanish flu in 1918, 1919. And then there was one in 1957, another one in 1968. And here for the 21st century, it was the pandemic flu of 2009 with H1N1. So these are viruses that we continue to watch. And currently, there is really a signal around the circulation of the highly pathogenic avian flu H5N1. I will leave Stefan comment on it because he certainly knows much better than I do the surveillance of this specific pathogen and the risk associated with it. But I would say other categories of viruses that we would watch very closely are those responsible for viral hemorrhagic fevers. Ebola, Marburg, and we know that whenever the epidemic starts, I mean, teams are sent on site and participate in the early taking care of the patients, but also isolation, quarantine. And for Ebola, fortunately, now we have a vaccine that works well, so we can do this ring vaccination around the cases, and we are able to control them. So these would be 2 families of disease that I think we follow very closely, but the list could be going much further, and it would be just a bit tedious to try to visit them. But maybe I would now hand over to Stefan so he can tell us a little bit more about the situation with H5N1, which is really something that we are following very closely.
Yes, thank you, Arnaud. Yes, you are right. Of course, influenza viruses are really under surveillance in the veterinary field and human field. And in the veterinary field, when we had the cases of H5N1, a survey is organized around the farm which is infected to try to find other species infected, cats or other species. Recently, the virus have been identified in cattle and sheep in the US, for example. And so the idea is to try to see if these viruses are able to cross the species barrier and infect other species. But at the same time, we try to prevent the infection against H5N1. And I think France is the only country in the world where we are testing vaccines. We are testing 2 vaccines at the moment because there is international rules concerning exchange of animals that animals should not be seropositive. The objective was to prevent the importation of animal infected. But now things are changing to say, okay, if you are vaccinated, you are of course seropositive. This is the objective, but seropositivity doesn't mean you are infected. So at the moment, 2 vaccines are being tested at the moment with very good results. But at the same time, there is international surveillance of H5N1. All the isolates are sequenced, and we try to be sure that these viruses are not able to be humanized and to be transmitted as classical human influenza viruses. And there is also one virus which is, I think, and Arnaud gave information about hemorrhagic fever. It's the Crimée-Congo hemorrhagic fever virus, which is now present in Spain, which is present in the south of France, which is transmitted by ticks. So you have no clinical signs in domestic animals, cattle, horses, sheep, but this virus can be very dangerous, lethal for humans. So this is one, because there are many, one example of viruses which are under surveillance and for which epidemiological pattern has been totally modified this last decades, I would say. And we have also the same thing concerning diseases which are strictly animal, because one else is to compare the systems. You have pathogens which infect strictly humans, other which are zoonotic, infect both, and some which are strictly dedicated to animals. But I've got in mind, because I work in this field, foot and mouth disease, for example, and foot and mouth disease is a virus which is present in many countries and which is really a threat for the US, for Australia, for Europe. And we have an international network which is very similar to what you see in the surveillance of human diseases, to have as many information as possible to say to the countries, be careful, there is something appearing, so be prepared, or activate your surveillance systems.
Thank you. You talk a lot about viruses, both of you, and the course talks mostly about viruses. But besides them, can any other pathogens, such as bacteria or fungi, cause major pandemics? Or is it less of a worry than viruses?
I will start, and maybe Stefan will complete. What is important is to realize that among those emergencies, 2/3 are indeed viruses which come from the animal world, and they have crossed what we call the host barrier species, moving from animal to humans, zoonosis, as Stefan was explaining. So we are really most often talking about viruses, and as already mentioned, RNA viruses, because of their capacity for mutations. But there are bacteria that can give pandemics, and in the past, you may remember of the typhoid fever related to Salmonella. You may remember of plague that were responsible for very large pandemics. Currently, the one that is still very active in a pandemic mode is cholera. We see large outbreaks of cholera repeatedly, sometimes in specific context of wars, when you have a complete breakage of the sewage system and mixing of waters, and therefore risk for cholera outbreaks. And that was seen in Yemen, for instance, in the recent past. But you also see large outbreaks of cholera, which may eventually be linked with climate change, which is a phenomenon a bit complex that we are looking with scrutiny. But it has been documented, for instance, that the El Nino oscillations that you see in the Pacific can have as a consequences 6 months later accumulation of rain in the northern part of Bangladesh, and 5 months later warming of the ocean in the Bengal Gulf. I'm not sure about my pronunciation, but you may have understood, the Gulf du Bengal. And anyway, associated with it, 11 months later, cholera outbreaks. And if you shift by 11 months the El Nino oscillations, it's a remarkably well-fitting the cholera outbreaks of Bangladesh. So it's this long-distance phenomenon that we are currently capturing through a different type of surveillance and modeling. One other bacteria that I think we still have on watch is, I mentioned it, plague before, and you remember that it was responsible of very large pandemics. It had initially to be linked, with the presence of a specific rat, which is hosting the bacteria, and a flea, which transmitted from the rat to humans. But in some circumstances, some people can develop what we call pulmonary plague, and therefore first of all, it's a very severe disease. You die within 2-3 days in the absence of antibiotics, but also you become contagious by the cough. And in 2017, there was an alert in Madagascar where they do see in Madagascar every year cases of bubonic plague, the one that is related to the rat and the flea, in the highlands of Madagascar. But here, a patient had developed pulmonary plague, taken a small minibus, and gone through Antananarivo and started to transmit the disease by the pulmonary route, by coughing. And we saw, difficult to say the total number of cases, but it was in the few hundreds of cases of pulmonary plague. And that was very alarming because here you no longer needed to have the rat and the flea for the transmission. It was just contact person to person by the cough. And therefore, there was a very intense control measures which were taken in Madagascar to stop this outbreak. So just to tell you that there are sometimes bacteria that can be responsible for pandemics, and they are also being watched closely.
And just perhaps to complete what Arnaud said, is that concerning bacteria, which is not my specialty, so in the past or recent past, there are not new bacteria which has emerged as this is the case for viruses that nobody knew before. But you have, for example, the increase of antibiotic resistant mechanisms. So you see not specifically to one species of bacteria, but the fact that you have more and more bacteria which are resistant to antibiotics. I'm thinking to tuberculosis, for example, and the bacteriologists see more and more cases like that. So it's a phenomenon which is growing. So I don't know if it's a pandemic, but it's in the future, this will be a really important challenge in human and animal public health.
Thank you. You mentioned vectors with the ticks. So it brings me to the next question that was asked by the learners about the insect vectors. So which preventive actions are undertaken or could be undertaken to mitigate this risk of emergence through vectors? If you want to elaborate on that.
I will start with the mosquitoes, which I'm a bit more familiar with. Maybe, I don't know whether there are things to be told about ticks, and Stefan may have ideas about that. With the mosquitoes, there are many different mosquitoes that can be involved in transmitting different type of viruses or parasites. Of course, Anopheles for malaria, which is Plasmodium, a parasite. But you have also the Aedes families, with Aedes aegypti in tropical countries and Aedes albopictus in more temperate zones, also called the tiger mosquito. And those Anopheles will transmit what we call the arboviruses. There is yellow fever, you have chikungunya, you have Zika, you have dengue. And there are also diseases like Japanese encephalitis, and here it's more the Culex mosquitoes that would be involved. So you have a series of families of mosquitoes that can be transmitting a lot of disease. And according to some statistics, mosquitoes would probably be the biggest killers in the world, and they kill far more than what lions or hippopotamus can do, for instance. That's a small joke of something, well, if we can joke about that. However, the mosquitoes, yes, there are different ways of fighting them. If you take the example of Anopheles, for instance, of Aedes aegypti, for instance, which is responsible for, as I mentioned, yellow fever, dengue, Zika, or chikungunya, it's very important that you clean the places around you and whatever can contain clean water, because that's where the larvae grow. And it's one way by cleaning in your garden all the small pots that contain clean water that you will be able to limit the breeding of the larvae of the Aedes mosquitoes. You can, of course, protect yourself by using long sleeves, by using repellents. You can sleep. The Aedes itself tend to bite more in early morning and late evening, but it is changing its habits. But for other mosquitoes, like Anopheles, for instance, you would use a mosquito net at night to protect you. But now there are some new research techniques that are currently under development to fight those mosquitoes. They can be rendered sterile, whether by some genetic changes of the mosquitoes. You can do also irradiation, and there are some experiments currently done in the Ile La Réunion about irradiating mosquitoes to make them sterile. And you can also infect those mosquitoes with some bacteria called Wolbachia. And there have been some very interesting experiments in Asia, particularly in starting from a large trial performed in Indonesia, where they were infecting the Aedes mosquitoes with Wolbachia bacteria. And not only those mosquitoes do not reproduce as well, but also once they are co-infected with Wolbachia, they cannot harbor a virus, and therefore they can no longer transmit those viruses. So those new techniques are currently being experimented and could be quite promising in the future in helping in controlling the vector-borne disease, but here I'm talking about mosquitoes.
Yes, and just to complete what Arnaud said, there are more than 500 arboviruses which are described at the moment, with 150 which are pathogen for human. And I've just checked that the arboviruses can induce more than 700,000 deaths per year, so which is really important. And you have also diseases which are also spreading, like, for example, West Nile flaviviruses. And we see the evolution of these diseases in human and in animals also. So all the information that Arnaud gave you are the same also in the veterinary field. The only thing we have, I could say, because it's very complicated to control population of mosquitoes, is at the end to have vaccines, for example, to prevent the disease in human or in animals. In general, we have more vaccine in veterinary fields than in human. For example, West Nile, there is vaccine against West Nile in horses. There is no vaccine in humans, to my knowledge, which are commercially available, and for other diseases. But you have mosquitoes, you have also ticks, and we know that the area of ticks population is really increasing. The ticks can transmit different diseases, for example, Lyme disease, for example, TBV, which is a tick-borne encephalitis virus, which infect animals and humans. And there are very few molecules against ticks to control and to treat against ticks. Even in animals, you have very few insecticides. There is also one problem, which is the fact that these molecules can be dangerous in the environment. And in the animals, you have a very short period of time where it is active to prevent the ticks to bite. So there are very few things. So at the moment, there are some projects to develop vaccines against ticks. So the idea is to develop vaccines against the microbiota of ticks. And so when a tick will bite an animal or human, but animal for the moment, then you would have immune reaction which will kill the tick, or which will have consequences on the microbiota of ticks, which will die at the end. but this is really very preliminary results. And you have other vectors. I'm thinking to culicoides, for example, which are biting midges, which can transmit pathogens for human or for animals. And at the moment, there is absolutely nothing against these culicoides. You just have traps to check if they are present or not, but there is no way to control these populations. And I'm just doing some advertising. That's what we have built with one entomologist who is Annabella Fayou working in the Institut Pasteur, Xavier de Lamballerie, who is virologist in Marseille, and myself as a vet. I'm always the vet of the club, what we call Arbo France. And also now we have a European network, which is composed of all the people working in arboviruses, arthropod-borne diseases, not only viruses. So you have entomologists, you have bacteriologists, virologists, epidemiologists, modelizer, no? To try to develop research, because really we have very, very few tools against these diseases, which will be and which are already a threat to many, many countries, in particular because of the modification of the world temperature.
Thank you, Stéphane. You mentioned the spread of ticks. It was ticks that were more and more present on the globe. And there is a question in the Q&A that can relate to this. Like someone is asking if this increase in ticks presence just shows an increase. Do we already see an increase in the tick-borne diseases linked to, especially in northern countries, linked to the spread of ticks? Or is it just the fact that we see that we have more ticks, or?
No. We see we have more ticks. We see that the ticks are now present in areas where they were not present before. So this is clearly you have, when you do the cartography, I don't know if this is really English, but the cartography of the ticks dissemination, you see it is spreading, no question about that. And also the ticks can have pathogens, and you see also an increase of the diseases. At the same time, it's true that the surveillance is more active, more acute. So of course, if you try to find something, the probability you find it is higher than if you do nothing. I fully agree with that. But what is sure is the increase of the tick population in many countries.
Thank you. You mentioned already the One Health approach, and that was also discussed fully in the course. But some learners were wondering about the links that could be drawn between the One Health approach and the fight against HIV, tuberculosis, and malaria. So could you elaborate a bit on this link between the One Health approach and these three particular diseases?
I would say maybe Stéphane has a different opinion, but HIV, tuberculosis, and malaria are more examples of disease that now have become, for the pathogen that is currently responsible for the disease in humans, completely humanized, and they are transmitted from humans to humans for HIV and tuberculosis, and for malaria, you have the Anopheles vector that plays a role. But the animal that was at the origin of the pathogen that came to human is no longer involved in the cycle, between, as it could have been at the very beginning. For the most recent example, we know, for instance, that for HIV, the virus emerged probably somewhere at the beginning of the 20th century in Central Africa. And there are some reasonable evidence that it emerged from chimpanzees and for some subtype of HIV from gorillas in the southern parts of Cameroon and the northern part of what is currently the Democratic Republic of Congo. But now those diseases have become more strictly human, and therefore the fight is confined to how do you control its spread between humans and the curing, identification of patient, diagnosis, and curing. So this is a bit more the way it works. But it's really interesting. We have this seminar together with Stéphane, because we realize that there is a huge breadth of knowledge existing on the human side, and this is a bit more the sort of specialization I have, and on the vet side, and this is more Stéphane. And it's really difficult to get the bridge between these two worlds, so that they know how to exchange information, talk to each other, because clearly we know that some of the diseases that become humans come from the animal world, and we have to work very closely together. There are a lot of efforts in that direction. This is really what we call the One Health approach, but there is still a long way to go before we would be completely integrated, and basically that I would have knowledge, maybe not to the extent of Stefan, but reasonable knowledge in the vet disease, which I strictly do not have. And maybe Stefan would know a bit more about human disease, which he certainly knows, but maybe not to the same extent. And that's where I think we are really working to try to make these two worlds coming together. There are a number of initiatives, because it's really important that we work in this understanding of the transmission of disease from animals to humans and from humans to animals.
Yeah, just to complete what Arnaud said, and I fully agree with Arnaud, is that HIV is a very good example of, as Arnaud said, a virus which represents an animal, then were humanized and which now have human cycle and nothing to do with animals. But the One Health approach is that you have diseases which have both different components, animals and human. For example, West Nile, I'm involved in surveillance of West Nile, and we need to use the animal surveillance system to say, be careful, in this area you have West Nile virus. I'm just thinking about what has happened in New Occitania, which is near Bordeaux, or this area. So to make a surveillance of the blood transfusion, it was very important to know where the virus is or was. And thanks to the animal, birds and horses, the people in charge of blood transfusion knew that they have to select the area where they had to do some surveillance. So this is a very practical example. So both human and vets have to work together. And also the tools we use, the diagnostic tools, the vaccine tools, the animal models, the physiopathology of infections are very, very similar between human and vets. So we are talking the same language. Sometimes the diseases are totally different, of course. But if I just come back to what has happened with SARS-CoV-2, with COVID, this is something I have never really understood, is that in the veterinary lab, because of a disease which was blue tongue, which is a disease transmitted by biting midges, I was working on at that time and still, which emerged in 2006 in Europe, and we have many, many outbreaks of blue tongue. We have developed a network of veterinary labs with automats, which were able to do thousands of PCR per day. And so when there was this COVID situation, the vet labs were totally able to do PCR, because we know what is a PCR, although we are vets. And we had machines which were able to test many, many, many samples. And this is a good example of what should have been done to put together human and vet laboratories, because this is really the same strategies, the same problematics. I don't know if this is really English, but I think we have to learn from each side together. And for me, this is One Health. We don't have to copy, but to work together.
Thank you. There is a question in the Q&A section that goes back to highly pathogenic avian influenza virus, with like someone here mentioning the recent discovery of such virus in dairy cattle in some countries, and they are wondering if it's a jump of the virus over to another host, or is it just accidental? And is there a risk with that kind of jump to a new host? Is there a risk that it could result in a significant mutation that would pose a threat to public health?
To my knowledge, but I'm not a specialist H5N1. I know what it is, but I'm not working specifically on this virus. What they have observed in sheep and cattle is that there is no mutation. So it seems there is no specific adaptation to cattle. So probably, but I'm very careful, this could be an accidental observation. We just hope that this thing will stop, and there will be only some cases in cattle, and that's all. As I told you, the virus is the same, but it's true that H5N1 has already infected a lot of species, cats, foxes, cétacés, comment on dit, je sais plus, squirrels, forget the name. So many other species which are totally different from birds. And that's why we really need to have a very intense surveillance to detect a virus which would really have an important modification to change his epidemiology. But I think concerning the cases in cattle and sheep, that probably this is not anecdotal, but only infection without any consequences. But I'm very careful, of course.
I'm going to switch to another topic, but it was asked on the registration forms, but also on the Q&A section, talking a bit more about low-income countries. Could you elaborate a bit on the main differences between low versus high-income countries regarding the control of the risk of emergence and the control of emergence itself, with a particular focus on the challenges that are very specific to low-income countries?
So for low-income countries, I think they can play a crucial role in the fight against these emerging infections, particularly in the surveillance area. And it goes through the strengthening of surveillance systems and the transfer of capacity for allowing them to be completely autonomous, you know, in the way the surveillance is performed, which is already the case for many countries, because it's something that WHO has worked with countries for the past 20 years. But it is expensive. It requires good sentinel sites. It requires reference laboratories with equipment, trained people. So it's an investment that is necessary to continue. But it's really in the area of surveillance that I think at this stage they can play a major role, because they are a bit more exposed, sometimes because of the type of wild fauna that exists in those countries, some conditions of farming also that may facilitate emergence of viruses and crossing to humans. But it is absolutely essential that they get the support they need for the surveillance, because as we already mentioned earlier, it's really when you identify at the very early stage an outbreak that you can act and take the necessary measures for controlling before it spreads further. So there is an effort which is carried out at the global scale in strengthening surveillance systems in all those countries. And I think it is something that should absolutely continue under the new international health regulations. There are some more complex issues which are currently under debate. You know that there is a new agreement that is under discussion for the surveillance and response to pandemics worldwide. And this agreement will eventually be signed in the near future, but there are some very important ongoing discussions where low-income countries say we are, of course, ready to be here to help, assist, put all our surveillance capacity in identifying the emerging pathogens. But it is really important for us that we are not left on the side. If once you have identified the pathogens, once you have we have shared our all these first samples that we were able to obtain through our surveillance system, if a treatment is developed, if a diagnostic test is developed, if a vaccine is developed, we want to have access to those tools as diagnostics, vaccines, and treatment. And here there are some ongoing discussions, quite tense. You can read an editorial in The Lancet about 2 weeks ago by Richard Horton, the main editor of The Lancet, saying that currently the conditions are not there in making sure that would new emerging infections be diagnosed in the low-income countries thanks to their efforts. They would be playing this role of warning for the globe, but yet they are not guaranteed that they will have access to the diagnostic test, treatment, or vaccines would they be developed. And this is something we need absolutely to solve, because we have to be all playing together in the fight against pandemics.
And just one point to complete what Arnaud said, because this is exactly the same problems in the veterinary field. But there is something I'm quite proud we develop in my lab is to, as Arnaud said, develop diagnostic methods which are easy to be used in no income country. So we on foot and mouth disease, we develop lateral flow device. So in the country where this virus is circulating, you can very easily use this lateral flow device in a farm where you think the virus circulates. So you first of all have a result, negative or positive, with your lateral flow device. Then you could inactivate with citric acid your lateral flow device. You put it in a bottle, which is very simple and very not expensive. Then you send by post mail, because if you use the International United Nations system to send samples, it costs a fortune. So you just send by post mail this lateral flow device. As foot and mouth virus has a positive RNA genome, in my lab, we rescue the lateral flow device, the RNA, we put in cells, and we can rescue the whole virus. So this is, and now it is recognized at the international level, because at the beginning it was not easy. Some countries which were free of the disease say we don't want to receive positive RNA genome. So it was not too difficult to explain that if you send by post mail RNA, only RNA, the probability that this RNA will infect a cattle is quite very low. And so this is a very good example of having surveillance in the country, but also having very good diagnosis and having information about the strain, the vaccine which should be used according to the strain. So really this is in this direction that we have to go in future.
Thank you. There is one question that brings us back to surveillance, like in the Q&A section, but there are some questions on genomic surveillance. And could you explain a bit how it works and what can be learned from it? And the question was asked with a particular focus on viral hemorrhagic fevers, so Crimée-Congo or Ebola, for instance. So could you just explain a bit how genomic surveillance works in the particular context of these viruses?
Stéphane, do you want to go, or I can go? It's up to you.
No, you can go.
No, I would just take some examples, recent examples that we had with different diseases. I mean, for Ebola, for instance, what is important is that you know which Ebola strain you are dealing with, because they have different properties in terms of severity, and also we have a vaccine against the so-called Zaire strain, but we don't have a vaccine for all the strains of Ebola. So at the beginning, you need to know which strain you are dealing with. Later on, knowing exactly the evolution of the virus or whatever for an outbreak like Ebola, I would not be that at the top priority. I think it is interesting. I can tell you a little bit about how it is circulating. There are some very well documented map of how viruses have circulated in West Africa between Guinea, Sierra Leone, and Liberia during the 2013-2014. But I would say it's a little bit more for documentation, but it's not necessarily very important in the response to a disease like Ebola once you know exactly what Ebola strains you are dealing with. But for COVID-19, for instance, we all have seen how the virus was capable of changing. In fact, there were different modes of apparition of variants of COVID-19. There could be some mutations, there could be recombination, there were some transferred to the animal back to humans, but there were also, and that was the main mode of emergence of COVID-19 variants, this chronic infection in people who had immunosuppression. I'm talking here about people co-infected, for instance, with HIV. And when the HIV infection was not controlled, they could have COVID-19 infection that were lasting up to 6 months. And during those 6 months, you have an accumulation of mutations in SARS-CoV-2, which made new variants completely different from their predecessors. And Omicron is a good example of that. And therefore, we need to have this worldwide surveillance so that we are able to pick up early new variants, which themselves could be eventually more transmissible, more severe, or escaping to past immunity, whether that immunity is related to vaccines or to infection, natural infections. So for COVID-19, the surveillance, genomic surveillance has been extremely useful. And there were places that have been identified that could organize this surveillance and the sharing of data on this very large database like virological.org, I forgot the name of the main one, but maybe Stefan will help me. But anyway, I mean, there are some very large databases where all the viral genomes are shared almost live. And this information was very useful. In Africa, South Africa played a crucial role in the surveillance of the virus on the sub-Saharan continent, and Dakar, Institut Pasteur of Dakar, also played a very important role. So there were some places where genomic surveillance could be performed. And in the case of SARS-CoV-2 and COVID-19, because of the emergence of variants and their different properties, it was very useful to have that. GISAID is the name of the database.
Yes, I agree with Arnaud. If you are in an emergency situation, sometimes you don't need immediately the genomic tool, but it could be useful. For example, if I'm thinking to foot and mouth disease, which is a RNA virus changing very fast. You have 7 serotypes, you have subtypes, you have topotypes. So rapidly, you need to know exactly which virus you have to face. And so in this case, having rapidly the genome of the strain is very important. But you have diseases for which you don't necessarily in immediate answer, emergency situation, you don't necessarily need the genomic approach. It depends on what you want to do. But to have a traceability, to know where the virus comes from, or the pathogens come from, this is interesting tools. But once again, it depends on the situation. And also, it is important to use a good sample. We are doing such approach, but we sequence the virus directly from blood of the infected animal to avoid having mutation by passaging on different systems. So this is technology which has to be adapted to the situation you are working on with.
Yeah, thank you. There was another question about vectors, 2 other questions actually in the Q&A section about vectors and hosts. So the first one is, can bugs, so punaises in French, can be vectors of infectious diseases, and some that we should be particularly focusing on or not. And the second question is about bats, with some collectivities, urban collectivities, kind of like favoring the nichoirs. I don't know how you say how you say this in English, like the little houses for bats. And what do you think about this? So to favor biodiversity, but what do you think of this regarding everything we learned in the MOOC regarding the danger that bats represent for emerging viruses? So first, the bugs, the punaises, can they be vectors of infectious diseases to humans or to animals? And second question about the bats.
I'm sure there are tons of different bugs.
Yeah, but the punaise, I don't know how you
my very
translate
limited
punaise.
knowledge about bed bugs is that they don't carry pathogens, but I will let Stefan answer about bugs because you may know better. I can see that he's not more. I think what he's currently said about those bed bugs, you know, that if we talk about bed bugs, but I'm sure that it's a name that is too broad for all what it could entail, is that they don't carry pathogens that can be transmitted to humans at least. So that should not be a worry. Let's go to the bats, where it seems we are more comfortable for this discussion. The bats, I think it's really a topic of great interest. They are at the source of a number of diseases that have become pandemics and of very much significance. Clearly, with the beta coronavirus, whether we talk about SARS, MERS, or COVID-19, at the beginning there was a bat, and the rhinolophus bat for SARS-CoV and SARS-CoV-2 have been clearly identified. But you have other disease where we strongly suspect bats for Ebola, the food bats. It's clear from Marburg. It's most likely the case for Ebola. And we have also the Hendra virus in Australia, where fruit bats have been well identified, Nipah virus as well. So there are a number of diseases of importance where bats are seen. If I were to recommend one paper that has incredibly described the impact of deforestation on fruit bats in Australia, traveling to, they were looking at the situation of Andra. Andra is a virus that infects horses and eventually can get to human and give encephalitis, which are potentially fatal encephalitis. At the beginning, you have a fruit bat, and they have been studying for 25 years something that could be 50 kilometers square of an area of Australia. And they have shown how deforestation has displaced the bats, which during the winter time, which is between July and September in Australia, move to agricultural areas and to urban areas for food. And in agricultural areas, get in contact with the horses. And there is infection to horses and eventually transmission to humans. And all this phenomenon has been described in great detail. This paper published in Nature in 2022. I unfortunately don't remember the first author, but Rena Plowright, who is a very well-known ecologist working in Australia and many other places, was the last author of that paper. This is something I really strongly recommend because for me, it's one of the most achieved work which has looked at issues of deforestation and spillover of viruses to humans.
We just posted in the discussion section the link to the article that Arnaud just mentioned. So if you're interested, you can dig more into the subject of bats with this article. So I see that we have just a few minutes left, so I guess we could wrap up maybe with this last question. That's kind of a mix between what was asked before and now in the Q&A. That could each of you maybe give a few leads as to what could be done? I mean, what are the main points we should focus on to be more active in response to the risk of reemergence and to be more prepared at different scales from the governments to the public? And maybe particular thoughts on the currently drafted WHO Pandemic Preparedness and Response Treaty? And what are your thoughts on all of this and how would you like to wrap up this live session?
So considering we have 1 minute each, I will try to be brief.
Just a few leads, just a few points, the main ones.
For me, surveillance is key currently, because as we said, we can't predict where the next outbreak will take place, where and which pathogens will be involved. So surveillance is absolutely key. Rapid reporting whenever you detect outbreak is an essential component of the response. International collaboration here again is really important so that we can act extremely quickly on that. And when it comes to surveillance, I will probably hand it over to Stefan, because for me, I mean, the surveillance of the farming is, when we think about influenza, for instance, is really important with the risk of emergence of new viruses from poultry, pigs, reassortments, and whatever, and there are links they can have with wild bird and other animals bringing those viruses to them. And there is also the surveillance of the live markets, what we call the wet markets of Asia. When we look at the recent COVID-19 pandemic and the role that wet markets can play with animals that serve as intermediary host between those bats that we just mentioned and humans, the surveillance is key. Could also be that many of those animals could be just left quiet, live in the wild as they used to, and that's probably better for everyone. But if there were, for any reason, a need to have those markets taking place, they need to be watched very closely, because it's a clear source of emergence, and we have seen with COVID-19 how bad it can go. Stefan, the last word?
Yeah, I fully agree with what you said, Arnaud. Perhaps I would add that money for research is important. So I'm not totally impartial, probably, but I've just have an example at the emerging disease, which is episodic hemorrhagic disease virus, which has emerged in Europe. And for the moment, we have a lot of troubles to find facility to test the candidate vaccine, to do challenge, and this costs not a lot of money, but some money. And so if you compare the amount of money which would have to be put on the table just to have facility with the economic losses induced by this new virus, it has nothing to see. So I think sometimes you need to spend monies and you will save more money if you do that. And I think this is the same problem in human infectiology.
Do you want to add something, or is it the last word?
This is the last word for me.
Okay. So with this, I would like to thank you very much for your insights today. Thanks to all the people who were present for all the questions you asked before and during the session. We will try for the questions we did not have the time to address during the live session. We will try to get some answers for you and post them on the forum of the course on the FUN platform. The replay of this webinar, which has been, the webinar has been registered, so it will be accessible on YouTube in a few days, and an email will be sent to all of you in the upcoming days so that you can rewatch it if you want. And we would also like to remind you that the certifying exam, for those of you who have registered, will be available next week, so starting April 2nd until April 16th. And the deadline to register, if you haven't done so but wish to, is April 12th. So Clémentine, if you want to add something.
Thank you everyone, and thank you for the director, and to the director, and to you, Laurie, and good luck for the participants.
Bye.
Goodbye. Bye.